much for the introduction. Well, first and foremost, I would like to thank the organizers for giving me the opportunity to speak here, but also to come to the UAE for the first time. It's been quite mind-blowing. This challenges a lot of the ideas and preconception I had about this area in this country, and in a positive way. So I'm pretty sure it's not the last time I'll be here. And today I want to share a bit some past and current work that I've been doing around marine microbial interactions in the ocean. So every year, one of the most outstanding phenomenon occurs in the ocean, whereby organisms that are barely visible to the naked eye start to massively proliferate to form those blooms that you can actually see from space. Those organisms are called phytoplankton. These are unicellular eukaryotic phytoplankton, photosynthetic organisms that, even though they are small and come in quite abundance, also play quite a pretty important role for our planet, given that they are responsible for 50% of annual oxygen production on Earth, the other half being conducted by terrestrial rainforests. Even though they have this role, not only do they produce oxygen and capture carbon, they actually also really serve as the basis of the marine food chain, so you should see them kind of as the pasture of the sea. What has become quite clear in the past few years is that those eukaryotic phytoplankton do not live in isolation and instead establish numerous cell-cell interactions with other members of the planktonic community that can really impact cell fate. This can be with bacteria that live outside or inside those phytoplankton that can actually expand their ecological niches. They can interact with eukaryotic parasites or also viruses that can hijack the host metabolic machinery, leading to cell death. And they can also engage in other cell-cell interactions with ciliates that can really impact their ability to move through the water column. And even though those interactions happen at the microscale, given the magnitude of phytoplankton blooms in the ocean, this actually ends up having quite an impact on the way our ecosystem functions. That being said, there is currently really a lack of systematic understanding of the diversity of those cell-cell interactions in the ocean, and also how they actually may impact phytoplankton fate and function. And for me, 2024 has been quite an outstanding year in terms of discovery on that topic. First, early in the year with this paper in Science in which the scientists, marine microbiologists, were interested in studying bacteria that live inside algae in the middle of the ocean, which was actually quite, I would say, not such a hot topic by then, until they actually discovered that this is not any kind of bacteria living in an algae. It's actually a new eukaryotic organelle which fixes nitrogen, that they coined the nitroplast. And all of this started with actually looking at bacteria living in marine algae. A couple of weeks later, this paper from the MPI Bremen came out where they report the discovery of a rhizobia bacteria living inside a diatom. So rhizobia are the same nitrogen-fixing bacteria that live in plant roots. And here what was really interesting is that what they show is that this symbiosis actually solves a big puzzle about who were the actors responsible for nitrogen fixation in the North Atlantic. And putting those two papers side by side really makes me think that behind every phytoplankton symbiosis really lies an opportunity for, I would say, groundbreaking discoveries that can actually link topics ranging from the evolution of eukaryotic cell biology up to the way our ecosystem functions at a really big scale. And I guess this is what drives a bit research in my lab, where we are interested in the impact of those interactions on the fate and function of phytoplankton in the ocean, and through, I would say, three main questions. The first one is, what is the diversity and distribution of phytoplankton symbiosis in the ocean? By zooming in on specific case studies, we try to understand how those interactions can impact development of phytoplankton. And finally, in the current context, how are these interactions going to be impacted by climate change? To address this, we actually combine different scales of experimental setup. We try to push forward model system-based research in the very reproducible and controlled setup of the lab. We regularly conduct mesocosm experiments, which is basically a big flask, where we manipulate microbial communities at intermediate scale. And we, of course, regularly go out in the open ocean, and here particularly try to push our ability to study those interactions at the molecular and cellular scale, but directly in nature. And to illustrate this, I want to spend a bit of time talking about my postdoctoral work, where here the idea was to actually study the impact of host pathogen interactions across biological scales. The host is a unicellular eukaryotic algae called Gephirocapsa huxley. It's enveloped in this calcium carbonate cell wall around 5 micron diameter. This is one of the algae that is responsible for those massive blooms that you see from space. And those blooms are thought to be terminated by a viral infection by a giant double-stranded DNA virus, which is called EHV. And this virus is really the, I would say, primary mortality agent in those, or thought to be in those bloom demise. And back then, while trying to actually quantify the impact that a microbial interaction can have on an ecosystem process, we were facing a major methodological bottleneck, which was that we were completely unable to say how many cells were infected during a bloom. And as long as you cannot quantify the progression of active viral infection, it's going to be very hard to actually quantify the impact of viruses on the ecosystem and on the community. So to address this, I started in the lab by developing an approach which relies on labeling individual molecules of messenger RNA using in situ hybridization in single cells, coupled to imaging flow cytometry. So here, this is done in the lab in a controlled system, cultures of the host and the virus. Each row is a cell. The first column is a bright field image, and then we can actually specifically detect whether or not the cell is expressing viral messenger RNA, meaning that it's actively undergoing viral infection, or the host messenger RNA, in this case, which is a good proxy for the metabolic state of the cell. And because we did this through time and quite quantitatively in the lab, we were actually able to reveal new aspects in the life cycle of this giant virus. This was thought to be a lytic virus, meaning that the virus enters the cell, it replicates, and then the virions are released in the media through cell lysis. And what we discovered is that, in fact, very early during infection progression, there is a phase of the infection during which hosts are infected, but they start to chronically release, spit out virions in the media without killing the host, which is actually a very smart strategy in the natural environment because you could increase the viral load of the media without actually killing the host. That enables you to replicate. Because this was done with imaging flow cytometry, we could also track different cell phenotypes through time. And in particular, what you are looking at here is an imaging flow cytometry plot where each dot is a picture. And we are looking at DAPI, area versus circularity, and we could detect aggregates. Aggregates is a known phenotype during viral infection. Algae cells actually produce polysaccharides that induce cell aggregation and sinking to the bottom of the ocean. But what we show is that this could also potentially be a host defense mechanism because early during infection, those aggregates are really enriched in infected cells that can actually trap newborn virions and prevent them from being spread in the naive population. So this was done in the lab. Of course, the next step was to try and reach out to more, I would say, natural complexity. And we conducted a mesocosm experiment where the idea is basically to take 11,000 liters of natural seawater, to put it in a big flask, and to follow it through time. And here, the idea was really to try and follow the development and demise of a natural phytoplankton bloom, and in particular, to link the fate of individual cells, specifically viral infection, to ecosystem processes. Very long story, very short. We were able to apply this method in this natural community, and for the first time, to actually look at the progression of viral infection through time. So these are different days. This is only for one specific bag. And for the first time, we were able to actually quantify the progression of viral infection in that complex community. And for us, of course, this was super interesting because finally we had an assay that can enable us to quantify viral infection. But where it became even more interesting is when we tried to relate this to more, I would say, global processes, in particular linked to a carbon cycle. And for this, we used a specific measurement, which is called TEP. TEP stands for transparent exopolymer particles, or polysaccharides. These are sticky acidic polysaccharides that are produced by phytoplankton that induce aggregation. And aggregation is important in marine microbiology because this is what pulls down carbon to the bottom of the ocean. Here you can see the actual measurement of TEP concentration through time in the different bags that we had. We had 7. And there's a big challenge around modeling this. How do you model the accumulation of TEP in a very complex ecosystem? And we were trying different conventional methods. It was hard. We had a big error bar. And then at some point we said, OK, we know that infected cells produce more polysaccharides and that they induce aggregation. How about we try to inject those numbers into our models and see if we can better fit the data? And this is what happened. And that was actually quite good and interesting for us to show that or to highlight how important it is that when you want to actually model and estimate big processes like the sinking of carbon to the bottom of the ocean, you actually have to incorporate measurements of how many individual cells are actually infected during a bloom. So really to link what's happening at the single cell level, what's happening at the ecosystem level. We pushed that whole idea a bit further, and back then also conducted for the first time single-cell transcriptomics on natural marine communities. And back then, the hope was that we would be able to resolve and to discover new hosts of giant viruses in the ocean. When you do bulk metatranscriptomes in the ocean, you see that there's a lot of viruses. They are very expressed. They are super interesting. But because you are squishing all your biomass, you completely lose the identity of the host. And by using 10x, it took us 6 years, but we finally managed to actually resolve that new pair. And the last step of that journey was to actually deploy all of this in the natural environment. And I was lucky to lead an expedition using the Tarascuna offshore Argentina, where we actually used satellite imaging to really detect those blooms from space, and then actually take the boat, go there, and follow the same water mass for 7 consecutive days to actually try and now deploy all those tools, but directly in the natural environment. And this is still ongoing work. So fast forward, 2 years ago, I left the Weizmann and I started my own group at EMBL in Germany in the developmental biology unit. And while thinking about what the directions I wanted to go, one of the first things that popped up was that I really wanted to push forward the development of a good system to think about phytoplankton symbiosis across scales. And how do you do that? How do you pick a good model system? Especially as Omaya said yesterday, you know, the breadth of eukaryotes, and in particular microbial eukaryotes, is actually quite broad. And I decided to focus on a specific class of organisms which are called diatoms. These are unicellular eukaryotic phytoplankton that are enveloped in a silica cell wall. And in particular, to focus on one genus of that big group, which is called Chaetoceros, which I think is a really good system to think about and to study phytoplankton symbiosis. So Chaetoceros is a small square, basically, which has those spines, horns, setae. This is what gives them their name. There's around 400 species in this genus. Some of them are unicellular, some of them form chains. And it's an ecologically relevant organism given that it can form those massive blooms visible from space. But what makes it a very interesting system for me is that it's one of the rare eukaryotic phytoplankton that is known to establish cell-cell interactions with many organisms of the microbial community, with ciliates, nitrogen-fixing bacteria, eukaryotic parasites, viruses, and a given host-associated microbiome. We also have tools in the lab, many available cultures, some genomes and transcriptomes, scalable culturing, and in some specific species, some CRISPR. In terms of life cycle, Chaetoceros, like many other diatoms, has this kind of shoebox, very sturdy silica cell wall. It undergoes mitosis, and long story short, through time, as the cells undergo mitosis, the size distribution of the population gets smaller and smaller. At some point, the smallest cells hit what we call a sexual size threshold, which induces differentiation into male and female gametes. Those gametes conjugate and actually lead to an oxospore, which restitutes the initial cell size of the population. So it goes back to being a big cell again. And from a developmental point of view, it's actually quite interesting because the same monoclonal culture can have cells that differentiate into male and to female gametes. So in the lab, we've been also trying to enhance a bit the toolbox to study this unconventional model system. We collaborated with the recent Pan-European Trek expedition to establish a culture collection coming from all across Europe that we are currently phenotyping, but also, I would say, pushing for more advanced tools like single-cell transcriptomics, live imaging. What you see here is actually just a cell that divided. You're seeing it in a loop. And also, as you heard from Omaya yesterday, we are currently preparing a manuscript where we report conducting expansion microscopy in diatoms, in particular in Chaetoceros. And for us, this is amazing because suddenly we can actually do antibody labeling, which we couldn't do for a very long time. And this also really opens stunning perspectives. So for the rest of this talk, I actually want to focus on one aspect of what we do, which is mostly related to what we've been doing in the natural environment, and starting with our efforts in trying to develop tools that enable to chart in a systematic way that diversity of interactions in the ocean. So in marine microbiology, we've become very good at making lists of species very fast, and genes and genomes. I can go now in the ocean, take water, I sequence it, and I can see what's there. But we are still very bad at understanding the complexity of interactions that happen between those different species. And so that was really the main goal of this project, which is actually a continuation from my PhD, is how can we accelerate the discovery of symbiosis in those environmental samples? A main challenge that you can see on this picture is that we are coping with extreme morphological diversity in our samples. This is a planktonic samples of organisms between 20 and 200 micron. You have all the shapes and sizes, all the different types of silica cell walls, calcium carbonates, strontium sulfate. This is a complex environment. So what we've been trying to do and what we aim for is to actually now harness the power of AI to really conduct whole size spectrum plankton sorting, which is completely size independent because, of course, one of the easy answers is to say, let's do cell sorting. Most conventional cell sorters cannot cope with that level of diversity and sizes. So what we've been using is actually a combination of what is called feedback microscopy and photoactivation. So photoactivation is an approach that I've done in collaboration with Rainer Peppercork, who relies on a cage dye, which doesn't emit light by default. And when you excite this cage dye with a 405 laser, the cage dye opens and it starts to emit light in the green channel. So what we do is that we take our plankton sample, we cover it with this cage dye, which is actually conjugated to polyl lysine, and this is what sticks to the cell surface. And then under the confocal, we use AI to actually recognize our diatoms of interest. And then, still under the confocal, we tell the confocal to specifically excite that region of interest, so much so that the diatom turns green. So you're really lighting it up like a Christmas tree. What is interesting is that it's not only the diatom that is lit up, it's also the diatom and all its associated organisms, bacteria that are living inside, viruses, ciliates that establish cell-cell connections. So you're not actually labeling a single organism, but a kind of mini holobiont. So what does it look like? We actually start by scanning our planktonic samples. All those small boxes are what we use to then classify our organisms and identify the ones that we like. And then we specifically photoactivate, in our case, Chaetoceros. So this is exactly the same cell before and after photoactivation. So the signal is actually quite strong. And then thanks to this signal, you can actually then bring it to a cell sorter. And now you can actually sort what you're interested in, which are cells that have been photoactivated. And I think it's important to highlight that for us, this is a main difference because this is not a GFP positive cell. This is not something that we've transformed, right? It means that you can, with this approach, actually pretty much label any organism of interest, but without having any prior, I would say, molecular tools to actually label it. Once this is done, we actually currently move to single-cell sequencing. Right now, only metabarcoding, which is an approach that basically resolves the taxonomic identity of organisms that are co-isolated. And of course, this generates a lot of data, so we try to, as much as possible, really deploy pipelines that are reproducible and scalable. So we are at a stage right now where pretty much all of those parts talk to each other and work. And now we are entering the phase where we are starting to apply this pipeline to what I call a treasure trove, which are biobanks of samples that have been collected over the past few decades. We currently hold over 15 years of planetary scale sampling that originates from the Tara schooner, which cover pretty much all parts of the ocean. And we know, or at least I know from my PhD, that these samples are really holding a lot of secrets that we still have to uncover, but we didn't have the tools until now to do it. And I think now is a very exciting time to do so. And more recently, with this pan-European Trek expedition, we actually know that some of our locations also hold new symbioses that need to be characterized. And here it's very preliminary because we don't really have the data yet, but I want to show you a bit where this is going. And the idea is obviously to rebuild some networks of species-species interaction at scale, in which nodes are organisms, in our case, diatoms and associated species, and the links between those nodes are really physical interactions. What does this change compared to what we did before? Well, first of all, we can provide ground-proof experimental validation of co-occurrence networks that are usually conducted with more computational and network inference. More interestingly, we can actually discover new symbionts and their prevalence in the ocean. We can further discuss and think about selection mechanisms of host-associated microbiomes and their coevolution. And finally, what is important is that those networks are not, they are actually individual networks that originate from different locations in the ocean, right? Because we can do this per geographical location, which means that these come from different environments. And what we can do then is actually think about how those different community structures are sensitive to environmental change. So the long-term vision is really to go towards an atlas of cell-cell interactions. Of course, we focus on chaetosaurs, but the idea, and it's built in a way that it can be expanded to other organisms, to address different questions related to predator-prey, to other downstream assays. We do right now single-cell metabarcoding, but of course the idea is to push those frontiers as the method will be deployed. For the second part, still thinking about what we can do in the natural environment when we don't bring stuff back into the lab, is what happens if we try to really zoom in on specific interactions and try to study that process a bit more in time. The first part that I showed you is a huge spatial scale, but as you may have noticed, this is a one-time snapshot sampling in the ocean, and it's not really a process that we can follow. So how can we zoom in on specific interactions? And for this, I need to introduce a bit the idea of motility in diatoms. Most of the diatoms that live in the ocean are not motile. They do develop a flagella at specific times of their life cycle, but most of their life is actually non-motile. And this comes at a cost, which is that as a non-motile organism, you will sink out of the photic zone, which is actually quite annoying when you are a photosynthetic organism. You cannot really explore different nutrient spaces when you are in nutrient-poor waters. You cannot have a flight mode facing predation, and you also have limited dispersal in terms of population. Diatoms have found a way to become motile, not by evolving a flagella, but by establishing cell-cell interactions with motile organisms such as ciliates. It has been hypothesized, though not proven, that some of the benefits of such interaction, so this is a diatom, and this is actually a Vorticella ciliate. It has been suggested that this interaction, the flow generated by the ciliate, is actually renewing nutrients in the diffusion boundary layer of the diatoms and increasing the uptake of nutrients, hypothesized but not proven. And I showed quite a while ago that it could also serve as a potential armor. So here what you have is actually a diatom, which has a ciliate in the middle, and we know that those organisms are actually quite prevalent. So we see them, we can do some stuff with them, but the problem is that ciliates are very hard to culture. It's impossible to bring them back into the lab. And so what we tried to think was kind of a reverse approach of saying, well, if you cannot bring the ciliates in the lab, why not bring the lab to the ocean? And here I want to introduce the TREK expedition, which I was lucky to coordinate. TREK is an acronym for Traversing European Coastline. It was an 18-month expedition whose goal was to explore the impact of terrestrial ecosystem on marine ecosystem all along the European coasts, and was a tight collaboration between the French vessel Tara and the EMBL. One specific aspect I want to highlight from this expedition is what is called the Advanced Mobile Laboratory, which is this huge truck whose goal was really to advance our ability to conduct molecular and cell-level analysis on complex marine samples. It's important to understand that when you're on a vessel like Tara, you cannot use a confocal. You cannot, it's hard to put a sorter. It's hard to preserve samples to do electron microscopy. So here what we try to do is to bring that technology to the coastline to try and really push what we can do with our marine samples. So the Advanced Mobile Laboratory has those two massive panels that open in the middle that lead to a lab of around 40 square meters. This is in total 12 meters long, and it's equipped with sample prep for electron microscopy, high-pressure freezers for you that know, confocal microscopy, but also imaging cell sorting. There's actually a recent piece that was written on it in Nature if you want to scan the QR code. But in general, my lab has been focusing on those very few days in the expedition where the truck was side by side with Tara, whereby we could actually, in 4 hours, get a full description of the marine microbiome thanks to what Tara has been developing for now 3 decades. But at the same time, I could focus on my organisms of interest and really conduct targeted molecular and cell studies. And this is what we've been doing by harnessing image-enabled cell sorting directly on spot. So When you sample a boat like Tara, on a boat like Tara, as I mentioned, you have complex communities, and this actually prevents you from conducting a certain number of downstream analysis. And we've been harnessing the presence of this imaging cell sorter to move from samples that are actually pretty dirty, pretty complex, full of sediments, to something that nearly looks like a monoclonal culture. We've been specifically targeting, during those 18 months, our key player, which is Chaetosaurus. And this combined purification and concentration live of key players can really enable us to conduct advanced assays like single-cell transcriptomics, behavioral assays, but also sample prep for electron microscopy. And the nice, I would say, case study, again, this is super preliminary, but I thought it's nice to share, is when we bumped into this novel symbiosis between diatoms and ciliates in Estonia 18 months ago. So coming back from one of our sampling expedition, we look under the microscope and we were actually hoping to bump into such an interaction. We discovered that we had a symbiosis between Chaetosaurus in the middle and peritric ciliates that are actually attached to it in a mechanism that we still don't really understand. We processed this through imaging cell sorting, discovered that we could actually easily differentiate free-living from symbiotic units. And one of the first things that we decided to do after this sorting was to first quantify. So what we see is that over a couple of days, around 15% of our diatoms are systematically associated with a ciliate, so quite prevalent. And on the ciliate side, we see that 100% of the ciliates are always attached to diatoms. So quite prevalent, but also specific. We do not see any other diatoms with those ciliates, and we don't see any other ciliate on those diatoms. And there are other phytoplankton in that community, right? But it was somehow very specific. Mechanism and selection that we still don't really understand. And it's also an interaction that makes diatoms motile. Here is a confocal live video where we see the Chaetosaurus in the middle and the two ciliates on each side that are really moving this diatom into the water column. And we've been collecting a lot of movies and now trying to better understand what's the impact of that symbiosis on the behavior and displacement of the two organisms in the water column. Of course, now that we are in the field, one of the main advantages of this mobile lab is that we could sort a lot of symbiosis and we could actually try different culturing media. And one of them actually ended up enabling us to keep them in the lab long enough to actually record the whole life cycle. And one of the first questions was, how do cells establish symbiosis? What you see here is a Chaetosaurus cell, and actually this is a ciliate. It's actually two ciliates because it divided. It underwent binary fission. Those ciliates are encapsulated in a lorica, which is kind of a proteinaceous cell wall. So they divide. We were very excited. That was in the lab. And at some point, one of the daughter cells gets expelled. It gets expelled without the lorica. So it's completely, it's basically a ball with a lot of cilia around it. It gets expelled. It explores the space for quite a while. And at some point, after what seems to be a really strong behavior, basically picks a diatom. It picks a diatom, and within a couple of hours, it's actually completely rebuilding its lorica. That whole process takes around 5 hours. The last step from 3 to 4 takes around 1 hour. And this is what you see here. This is exactly the same cell. And this is actually the process during which here we have the diatom, and here we have actually the ciliate that is building its lorica. So this is the time lapse. And what we were very interested in is exactly this area here. How do those cells actually build such a sturdy bond with a unicellular eukaryote in such a short time? And to conduct this, one of the ways we tried to figure out what's the cellular process that mediate the establishment of the interaction, we of course tried confocal microscopy. So this, for example, is the ciliate. This is the diatom. Unfortunately, expansion microscopy did not work on this symbiosis, so we thought we needed to go a bit further. And one of the advantages of being on the AML, the Advanced Mobile Lab, is that we could high-pressure freeze the samples to conduct electron microscopy and really dive specifically on that cell-cell connection. This is like quite fresh from the oven, so to be honest, we haven't finished understanding what we are looking at. So if any of you actually has some insight, I would be really happy to hear. What you have here is the diatom, this is the ciliate, and you're really going to slice through that interaction, focusing on that junction. So here we are slicing through it. This is the lorica of the ciliate, so this proteinaceous cell wall. This is actually the chloroplast of the diatom. And here we are hitting, slowly reaching this kind of junction. We do not know what this material is. We don't really know what is the mechanism of secretion to build that material. Here you can see structures that are reminiscent probably of cilia, of microtubule, maybe here basal bodies. And here, with this image, what we are trying to do now is to understand what is the sequence of events, what is the mechanism by which this ciliate is actually building this bridge. And it's actually quite interesting in terms of bioadhesion, because in 1 hour, those organisms managed to build something that is quite irreversible in a salty, wet, and very unstable environment. So I also think that there's some potential interesting application there. Last but not least, this was in Tallinn. This is in one spot. How prevalent is this in the natural environment? And by teaming up with Tara and some high-throughput imaging approaches, we were able to show that it's actually found in all the Baltic Sea, which is a very nutrient-depleted space, at least at the time where we were there. And so this distribution actually raised the hypothesis that maybe it is also a nutrient-mediated symbiosis. And the way to test it, the way forward, is going to actually be to process single-cell transcriptomics plates that we have, to look at the induction of transporter genes. But also, later this year, my student Thomas is going there again to try and do some incubation directly on spot. So I'm running out of time. Just a few take-home. We are trying to establish Chaetosaurus as a relevant system to think and study symbiosis in the ocean. That today we can harness AI and different light microscopy approaches to really target planetary scale inventories of symbiosis in the ocean, but it will always boil down to really going deep into specific interactions, and we try to do this now directly in the natural environment and not necessarily be limited by what we can bring in culture or not. And I just want to finish by advertising an upcoming seminar, which is a bit related to what I've presented, upcoming EMBO Symposium on the Wild Frontiers of Model Organism, and if you want to know more, I'm happy to share. That's it. Thank you very much.